Thank you for inviting me to speak at this meeting. It's an amazing meeting. A couple things. First of all, I tend to pace, so if I move away from the microphone, just jump up and down or do something to remind me to come back. And second of all, I'm going to go through some things that are already published. They're a little older. If you've heard it before, please feel free to take a short nap. Okay. So for those of you that are in the cancer field, you know that in 2000 there was a landmark paper published, Hallmarks of Cancer, that was basically gene-centric. All of these things were described in the context of various gene mutations, changes in gene expression, et cetera. And then in 2011, metabolism hit the big time, and they've added hallmarks, including metabolism, and those of us working on brain tumors all danced. So this has really led to, and actually Dr. Tom Seyfried is going to speak later in this meeting, is clearly a leader in this field, the idea that, well, cancer was thought of as being a genetic disease. Maybe it's a metabolic disease. And in our laboratory, we call it the perfect storm. And the reason for that is we know genetics has a place. There's no question there are mutations, there are changes in gene expression. We know metabolism has a place. There's no question about that either. And now we know that epigenetics has a place, as if it wasn't complicated enough. And these things all interact. And I think in cartoons. So this is what I think it looks like. And that's what happens when you start to study cancer. You can pull out any one color of yarn, but it doesn't unravel the whole mess. So all of these things are totally and completely interacting. So when you look at some of the metabolism issues, and I just circled a few of them in red, all of these things all around the hallmarks wheel, essentially, really have metabolic issues. So that's why we call cancer the perfect storm in our lab. So it has to do with the genetic makeup of the individual. Physicians in this room, or anybody who's ever been a patient, you go to your doctor, they ask for your history. That's because your genetic makeup has something to do with your susceptibility to various diseases. There are environmental effects on cells. There are the ones we can control easily: smoking, sunburn, chemical exposure. May or may not be easy depending on whether you know about the chemical already. And then, of course, the issue at this meeting, which is diet. Some of that is just lifestyle choices, and some of it, honestly, could even be environmental and ethnic, in terms of what certain ethnic groups tend to eat. But then there's the two that I think are really possibly most important, and that is stuff happens, okay? As we age, there are random cellular changes in our bodies. Now, we can accelerate or decelerate them with some of these things, but some of these things are going to happen no matter what you do. And then once a tumor starts to form, a lot more stuff happens, okay? Cancer is Darwinism speeded up. The cells are genetically unstable. They're shifting things all the time. So all of these things is why we call cancer the perfect storm. And that's why in our lab the goal is to treat it any way you can, all right? And that includes to target rapidly growing cells. That might include chemotherapies, radiation. Target molecular changes. If you find a molecular change and you find a drug that'll hit that particular target, go for it. Enlist the help of the immune system. That's a big thing in cancer research these days, immunotherapy. And then our favorite, which is to target weaknesses due to aberrant metabolism. So in the very beginning, what we did was we actually just took cells in culture, and this was a cell line from a patient. This was their 4th tumor. This young person had had everything done to them that could be done, and this was his 4th surgery, and a colleague of mine made these cells. And they are very autocrine. That means they make growth factors, they spit it out, they suck it back in. So basically, it's a cell line that feeds itself. And this is what we found. If you just look at the growth of the cells, that's the line you get. If you look at BCNU, which was the drug of choice for brain tumors before temozolomide, this is a drug that this patient had been given. The cells lag a little bit, then they start to grow. That's pretty much what you see. When we added ketones, it slowed the growth of the tumor. You can see the slope is lower. And when you put the two together, it wiped the culture out. And when I saw that, I said, okay, now I'm willing to start working on this because it can be done along with other therapies. The name of the cell line is AO2V4, which is meaningless to you guys. But what is meaningful is we did not drop the glucose in this cell growth media. So it has full glucose, which is pretty high. And then we did 5 millimolar each beta-hydroxybutyrate and acetoacetate, and the drug is a clinically relevant dose of BCNU. Clinicians might recognize it as carmustine. So we then went into a mouse model, and basically we got the firefly gene, LUC2, put it into mouse glioma cells. They are bioluminescent. And our experimental protocol is we inject these cells into the brain of albino C57 black 6 mice. It's an immunocompetent model. The cells are bioluminescent. We've been using Ketocal as our ketogenic diet because we feel it's more like what a human can get as opposed to using an animal diet. It's nutritionally complete. We just mix it 4-to-1 with, it's a 4-to-1 ratio of fats to carb plus protein. We mix it in a 2-to-1 paste. The animals like it, they eat it, they don't lose weight. Sometimes they get a little greasy when they roll in it, but they're okay. And we let them eat as much as they want. I'm not sure why this seems to be doubling, but 2 slides at once. But the animals can eat whatever they want. So on day 0, they get surgery. In 3 days, they're pretty much recovered. The tumors are scanned. They're randomized to a treatment arm. We do blood glucose and ketone measurements. And in this particular model, I'm an animal lover, so this is kind of important to me. The animals have a very reproducible set of symptoms approximately 24 hours before they're going to succumb. So when we see the size of the tumors getting big enough, they're checked multiple times during the day, and they're humanely sacrificed. And that also allows us to analyze the tissue. This is what the imaging instrument gives you, but it will give you a quantitative readout. It looks like she's awake, but she's not. Mice don't have eyelids. So it will give you a quantitative readout of photons in this animal. And again, this is older data. It's been published, so same as what other people have found in similar models. Here's the animals succumbing with standard diet, ketogenic diet. It's significantly increased. When we gave the animals temozolomide, again, increased. But when we did it with radiation, this really blew people's minds, including mine. You got an increase in survival over no treatment, which you would expect. But with Ketocal, we actually got the tumors disappearing. One thing I will mention with respect to caloric restriction, we do not calorically restrict. And the reason we don't do this, well, the main reason is I knew the only way I was going to ever get a clinician to do this is if I didn't even mention the word restriction. So we don't do it. This particular animal calorically restricted herself. I have no idea why. She dropped about 20% of her body weight. Maybe she looked in a mirror. I don't know. But for whatever reason, she calorically restricted herself. We did not euthanize her because she seemed totally healthy. There was no indication that there was a problem, and she did live significantly longer. But she did eventually succumb. Just to prove that the animals did indeed have tumors, here's one that got radiation alone. Here's one on Ketocal plus radiation. The tumor did disappear at day 101. We image every 3 days, so that's why it's a weird number. We switched them back to standard diet, let them run around for another 200 days. The tumors never came back. There was no evidence of any viable tumor at necropsy. So how does this work? We started looking at things, and because we were working with Dr. Zhang Ro, he's my keto mentor, and they looked a lot at reactive oxygen species. That's one of the first things we looked at. And we have a compound, or you can purchase a compound that shows reactive oxygen species. And what we found was that the ketogenic diet reduces reactive oxygen species basically as early as 24 hours after you start the animal on the ketogenic diet. Now we have a little confusion. Radiation works to a large extent through the production of reactive oxygen species. The ketogenic diet reduces reactive oxygen species, but it seems to really potentiate radiation. So it took a lot of years for us to start to come up with a possible answer for this, and hopefully at the end of this talk I'll kind of give you where we are with that. Again, looking at things from the epilepsy community, that gave us some good hints, looking at growth factors. Growth factors are very important in brain tumors, and insulin-like growth factor, green fluorescence in standard diet, and I don't know if you can see it with the lights, but the blue are just nuclei, and you can see that there's a definite reduction. We also have reason to believe that the signaling pathways on other growth factors are probably dialed down as well. I should mention that a lot of the things we looked at, pharmaceutical companies are coming up with drugs to do these things. So for example, turning down growth factors, there are a fair number of drugs in the brain tumor and cancer in general where they're trying to dial down growth factors. Inflammation. So This machine also allows the use of various fluorescent markers. This is a fluorescent marker for cyclooxygenase 2, which is a main player in inflammation. And you can see that the ketogenic diet has a reduction in COX-2. There is an increase after radiation, which you expect, and the ketogenic diet reduces that. And again, from the point of view of wanting physicians to accept this for patients, this is really nice. We found a reduction in edema. So the edema around the tumor is clearly reduced. Again, really important, because what kills an animal or a person is not just the tumor, it's tumor burden. Tumor burden is the tumor, and it's also edema, because it's got no place to go, all right? So that pressure is a problem. So if you can reduce the edema, this is a really good thing. We found a reduction in angiogenesis. I know Tom has also shown this in his model, so it's nice when you see the same thing in different models. The green is just CD31. This shows new blood vessels. Looking at 10 random microscope fields for how much green is there. Definite reduction. We also looked at hypoxia. So there is a compound or a molecule called hypoxysense that's commercially available. It detects a marker of hypoxia. Again, we could do it in vivo. And you can see there's a definite reduction in hypoxia. Just looking at Western blots, this is just amount of protein just to show why this might be happening. So again, this shows the same thing we saw with our in vivo imager, that there's a reduction in this hypoxia marker. A gene that's been very popular these days in tumor biology is called HIF1 alpha, or hypoxia-inducible factor alpha. It turns on a boatload of things, and hypoxia turns it on, or hypoxia causes more of it to accumulate, I should say. And indeed, if you just grind up the whole tumor, do a Western blot, there is a very definite reduction. There's also a reduction in another gene that, again, turns on a boatload of things. So one gene usually turns on a whole cascade of things. And this is another one that's important in tumors called NF-kappa B. Total NF-kappa B doesn't matter. It doesn't work unless it's activated. So the total doesn't change, but the activation clearly does. Then we started collaborating with a laboratory at Arizona State University on immunology and the immune system. Like I said, anti-cancer immune issues are a big thing these days. And we started looking at this, and the thing, I'm not an immunologist, so the thing that we actually ended up looking at is this here, PD-1, PD-L1, and CTLA-4. Those are two molecules that are at two molecule pairs that are actually involved in inhibiting the anti-cancer immune response. And there, again, pharmaceutical companies are coming out with inhibitors for those. This is published, so you can just kind of get the paper. I didn't put the paper here, sorry. Ah, I put it there. So it came out, I think, last May. So basically, what did we find? We found that the animals on ketogenic diet had a decrease of PD-L1. They had a decrease of CTLA-4. Pharmaceutical companies, eat your heart out. They decreased one of these cytokines that, again, is part of the inhibitory cascade in tumors. It increased some of the anti-tumor cytokines, and it increased tumor cell killing by CD8 plus cells. So in addition to possibly helping all these other therapies work better, we think that the ketogenic diet is likely to help some of these new immunotherapies work better too. So in order to get to mechanism, we went back to our cell culture roots, and we started just looking at the effects of beta-hydroxybutyrate on cell growth, radiosensitivity, and then that lets us get into mechanisms a little bit. And again, our very first experiment, is why we've actually focused on looking at ketones only. I am not suggesting that the glucose isn't important. I think it's very important, but you got to start somewhere. So we are starting just with ketones without changing the glucose. And then once we get some things really nailed down, of course, we'll go play with the glucose. First thing is to show that what we see in the animals is recapitulated in cell culture, and it is. These are the cells that we use in the animals, the bioluminescent ones. In all cases, black is control, red is radiation, and in this case, we actually only use 2-gray radiation, which is a kind of low dose. And then we use 5 millimolar BHB, and then we put the two together. And the important line is the green line. at every point it's lower. So even here with 2 millimolar BHB, that's not enough BHB to affect these cells, but it is enough to enhance radiation. Up it to 5, it's enough to enhance radiation. Up it to 10, and basically it really works. We also entered into a collaboration with Dr. Brent Reynolds, who graciously shared some of his glioma stem cells with us. For those of you not in the brain tumor field, glioma stem cells are are now thought to be cells that are very important in tumors because they tend to be found in areas of hypoxia, and they tend to be resistant to radiation and resistant to chemo, and the thought is that they help kind of cause tumor recurrence. So this experiment, we just take the cells, put them in culture, treat them with beta-hydroxybutyrate, radiate them, and then they grow in these little spheres, these little clumps, so then you break the clumps apart and you count the cells. And with one of them, 5 millimolar beta-hydroxybutyrate clearly reduces growth, radiation reduces growth, but the two together, again, they potentiate each other. This is from a different patient, a little bit more sensitive to ketones and really sensitive to beta-hydroxybutyrate plus radiation. So for the people in the brain tumor community, the minute you show radiation resistance and chemo resistance, they say, but what about the stem cells? Stem cells work too. So again, getting back to that whole perfect storm thing, I think you really do need a pluripotent approach. You need an approach that's going to affect a lot of aspects of the tumor because tumors are really, really smart. You come up with something to block a cell, and the cell next door says, yeah, I can do something else. And that's how these tumors do tend to come back. You can kill most of the tumor cells, but the ones that are resistant to what you're using come back. So the ketogenic diet affects a lot of things on this hallmark wheel, and in fact, I am willing to bet that we're going to find that it affects everything. So what could possibly be a mechanism that causes this many changes? This was a big question we had because the NIH, of course, said, show us the mechanism. And I said, which one? And we've kind of stumbled on one that might be pretty important centrally, and that is epigenetics. Epigenetics is very popular. It's a big buzzword. It's a way you can change gene expression where you're not actually changing the sequence of the DNA. So we're not talking about mutations or anything like that. It turns out, some papers have been put out that beta-hydroxybutyrate is actually an effector of epigenetics. It's called a histone deacetylase inhibitor, or an HDAC inhibitor. And in fact, HDAC inhibitors are now being looked at clinically in some clinical trials. The problem is they're generally very, very non-specific. So there have been some toxicity issues and things, but they are being looked at. Yet one more thing that the ketogenic diet does that the pharmaceutical companies are looking at. So just very briefly, I'm not an expert in this, but histone acetylation, histones are the little proteins that the DNA is wrapped around. And when the histones are acetylated, it suppresses tumor suppressor genes, it lets the cells grow faster, and basically it lets the DNA get all screwed up. If you get rid of those, you open the chromatin. Now you turn off tumor suppressors, DNA damaging agents work better because the DNA basically is open. And I think in cartoons, so I think if the DNA looks like this, you can't really get anything in there. But if it looks like this, hey, you can do all kinds of things. And it also helps increase cell cycle arrest, increase apoptosis. These are all, for those of you who aren't in cancer, this is all good. Histone deacetylases go that way. BHB is a histone deacetylase inhibitor. Now that was shown in normal cells. It also seems to happen in tumors. So this is just a marker for histone acetylation. This is just a loading control to show that we put about the same amount of protein in. These are two chemicals that cause histone deacetylation inhibition. And with no BHB, there's no acetylation. And with BHB, there's more. I don't have this quantitated because this is being repeated as we sit here, so it's kind of new data. But what we're showing is that BHB in our tumor cells is indeed a histone deacetylase inhibitor. And if you look at DNA damage, this is a marker for DNA damage. It just sticks to the end of damaged DNA. there is an increase in DNA damage. Now, this is not with radiation. This is just DNA damage from beta-hydroxybutyrate, which is interesting. Does that mean beta-hydroxybutyrate damages DNA? Not necessarily. We're still kind of trying to piece this out, but through normal things that happen in the cell, you actually will sometimes get some of this binding, and it might be that it's just not getting things fixed. We're kind of still working on that. But this being up in a tumor cell is a good thing for the patient. So what about the genes that repair DNA damage? These are very important for radioresistance and for some chemotherapy resistance things. Reducing all of these, this is again a loading control, reducing these means that cell is less likely to be able to repair the damage that radiation does. Here you go. BHB, in clinically relevant amounts, definitely reduces it. Again, very preliminary data. There's no question that we're repeating it, it repeats, but I don't have the repeats here. And just looking at quantitation, with BHB, all of these are down below 0. So now we're starting to get to a thought of, well, maybe this has something to do with how this is making radiation work better. Even if it's not changing the amount of reactive oxygen species, radiation has a lot of other ways that it damages cells, and the cells have to be able to repair the damage. Our normal cells can, that's why we survive. But maybe the BHB is blocking the tumor cells from repairing. The other thing we've done is enter into a collaboration with a really awesome collaborator at Imperial College in the UK, Dr. Nell Syed. We've sent her tissue from our animals, the tumors from animals on Ketocal and on regular diet, and she was looking at microRNAs. MicroRNAs are yet another class of molecules that are involved in regulating the expression of genes. And doing a global screen, she came up with a number that are very much increased. And sorry, I forgot to say something here. Come on. Well, I'll go to this and then I'll come. Sorry about this. There we go. Okay. The family of LET7 microRNAs are known to be involved in regulating the expression of an oncogene, a gene that causes cancer called MYC. This is not only important in some adult cancers, but it's very important in very aggressive forms of childhood medulloblastoma. So when we saw that these were up, we got pretty excited and we said, well, let's take a look at MYC because that's kind of a central player in a whole bunch of stuff. Son of a gun. MYC is way down. And finding pharmaceutical inhibitors for MYC has been somewhat difficult for pediatric medulloblastoma, so we're really excited that this might actually help in the most aggressive form of pediatric medulloblastoma, which is the MYC amplified ones. We've also kind of focused in, Nell and our lab has focused in on a microRNA 138 that's down here, meaning it's way overexpressed. You have to validate, big screens like this you have to validate, so she validated that yes, indeed, it is way overexpressed. It is usually down in a lot of cancers, including GBM, which means putting it back up is a good thing for stopping the cancer or slowing the cancer or changing something about the cancer that's going to be good for the patient. And it upregulates a bunch of things that are involved in downregulating it. In other words, having it low causes genes that are involved in therapy resistance to be up. So increasing this is going to reduce genes involved in resistance and therefore help sensitize. I know it goes back and forth. It's really confusing, isn't it? So what about clinical use? Well, there are clinical trials that were listed on NIHclinicaltrials.gov. There are published studies. But what's the bottom line of all this? This is some stuff that I put together for the meeting in Banff recently. And I actually spoke to the principal investigators on all these trials. And the bottom line is the clinical trials are really not mature enough yet to make any statement about survival. We all know anecdotal evidence. We all know some people in a trial, whatever, that have done very, very well. But in terms of what the clinicians want, 100 patients or whatever, it's really not quite there yet. The diet does appear to be tolerable. There have been no serious adverse events in any of the people that I spoke to. A few people do really, really well. A lot have average survival. But anecdotal information suggests that there should be efficacy in here. There is efficacy, and we definitely know more work is needed. Anecdotal evidence is not necessarily wrong. It's just evidence that isn't vetted in the standard clinical and scientific community. However, I will say if it's on Facebook, it must be right. So. Okay, quite a few of the patients we have are doing very well. We've got some in our institution, and again, people I've spoken to, they all have some patients that are just doing so much better than you expect. Like I said, if the social media says it's real, it is. But there are concerns that we as scientists and as clinicians and dietitians, who I consider clinicians, of course, really have to worry about. Comments like, while my patient was on the diet, the tumor stopped growing, and as soon as they went off, the tumor started again. Grandma decided the child needed candy, or something like that. Something very interesting from our institution, actually. There was a patient that had a 2-stage surgery. It's not surgery for recurrent tumor, but 2 stages, meaning the surgeon wants to go in kind of twice. They went in, took the tumor out. They waited a little while, and then they said, you know, the patient has no adverse effects. Let's go ahead and get the rest of it, okay? In between that time, and I don't remember exactly how long it was, but it was longer than usual in our institution. It might have been a few weeks, or maybe even a month. Usually it's a few days in our institution. The patient decided they wanted to be on a very strong ketogenic diet. I don't know if Andrew Scarborough is in the audience, but he's been watching your blog, and he said he wants to be the U.S. Andrew Scarborough. Quite a compliment, Andrew. And I saw the surgeon, and he said, you know, it's kind of interesting. I'm not saying it's the diet, but the tumor got smaller between what was left after surgery and the pre-surgery MRI for the second surgery. He said, this is really interesting. There's also been anecdotal evidence or anecdotal comments from neurosurgeons. I know a few in the UK, and again, one in our institution, that, you know, the tumor seemed a little less bloody than I would have expected. It seemed a little easier to get at than I would have expected. And then we've got patients that are long-term survivors. In fact, the patient that in our institution caused our clinicians to turn around and say, hey, maybe we should try this, is doing incredibly well. She was very, very strict on the diet. She was able to keep her glucose well below 100 even on steroids until she was weaned off the steroids. Her ketones were quite high. And again, the surgeons after the first few MRIs went, whoa, maybe we should try this. She's doing so well. And I love to hear that. What have we learned? I think we've learned a lot. I think mostly we've learned what we have to do. So we know about the tolerability, but when is it tolerable? When the patient is committed. I think dietitian support is absolutely critical. The patient in our institution, who was the first one that did really well, she actually touched base with our dietitian every day, even if it was just a, hey, I'm okay. They worked with the keto calculator every day. Physician buy-in, also critical. In our institution, the way it started is, well, I don't think it's going to make much difference, but yeah, go ahead, it's not going to hurt you. Until somebody did really well, and then a couple of them said, oh, wow. But a lot of times I get contacted, I'm sure a lot of people in this room get contacted saying, I can't get my clinician to go for this. They're telling me not to do it. They're telling me it's terrible. Think about it. If you were, God forbid, a patient, and you had to put up with this, your clinician is your lifeline. If your clinician is telling you this isn't going to work, and you're not going to like it, and you might as well go enjoy yourself because you're going to be gone in a year and a half, how easy will it be for you to pass up that glass of wine, piece of cake, drink, whatever? So I think that's really critical. Excuse me. There's lots of ways to do this. I think a lot of them are effective, but not in everyone. I think we have no idea which intervention is going to work for which person. It's similar in the epilepsy community, I think, where there's a lot of different ways to do it. But the epilepsy community has something that tumor people don't have. If it doesn't work, the person has a seizure. It's a pretty quick answer as to whether it's working or not. People ask in the cancer community, how soon will I see an effect? I don't know. Well, will it hurt if I have a piece of cake? I don't know. It's a lot harder. The quality of life, we did quality of life assays, and other people have too. It does not appear to be anything bad on the quality of life, despite what some of the clinicians seem to think, as long as the person's tolerating the diet. If the person thinks the diet is terrible, then of course their quality of life isn't as good, and they go off the diet, and then it's not a problem because they're off the diet. So for those who really kind of buy into it, it doesn't hurt quality of life, and it also doesn't hurt cognitive function, because we also have had some clinicians worry about that. The bottom, I think you've heard before, you're going to keep hearing, is funding for controlled clinical trials is absolutely critical, and funding for these things is insanely expensive. What do we need to do? I think we need to treat metabolic therapy like any other therapy. When we first started this, I was as much a culprit as anybody else. Well, it's just food. I didn't say that to mean it wasn't important. I said that so the IRB would approve the protocol, which, by the way, they did without a problem. But the problem is, if the patients are kind of thinking it's just food, they will basically not follow directions. So one of our patients, I remember, I don't deal with the patients, but I was talking to our dietitian, and I remember she was putting notes, and the wife's notes, the notes from the wife, the caregiver for the patient, is, he really hated this, he really hated that, and some relative said, this is okay on a ketogenic diet, so I gave him this. Well, it might be okay on a standard, you're healthy and want to drop a few pounds ketogenic diet, but that is not the same as a therapeutic ketogenic diet. And that's where the dietitians are critical. And because this isn't a pill, patients think they can listen to Aunt Jane, Uncle Joe, whoever, and say, well, they said this was ketogenic, so it's okay. It's like, no. This is actually a medical therapy, and we need it to be thought of as a medical therapy. Similar diets are not identical. We need to study the different results. We need to find out, why do some people, and I think the same is true in the epilepsy community. They've been trying to do this for quite a while, and funding, of course, being an issue, and they're way ahead of us, of course. What is the difference between patients on a 2-to-1, patients on a 4-to-1? Does it make a difference? Now, this takes lots and lots of people, which I guess in a good way we don't have for brain tumors, because it's not a super common cancer. But there are other cancers out there. We need to do more preclinical and clinical studies. I think we need multi-armed clinical trials. There are some trials like doing the modified Atkins versus some attempting for the 3.5-to-1 or 4-to-1, but maybe a multi-armed, multi-centered trial would really help. We are working on this. Other people I know they're working on this in the UK also, and we're going to possibly join forces. A registry of all the anecdotal patient data that includes physician information, so that anecdotal doesn't mean what does the patient remember about what they were told. I don't care how good you are. I don't care if you're in the field. When it's you or your loved one with this devastating disease, your memory might not be perfect. There are challenges to that, HIPAA being one of them. A lot of variability in what people are doing. Again, I mentioned the HIPAA reported versus patient care, but I really think we've got to start amassing this data, because there's so many people, most of you are probably in this room, that have this awesome data on a bunch of patients, and everybody has their own little set. But it's not enough to put it together, write a big paper, hand it to the clinicians and say, see, we told you. I think we need to motivate the patients, make the metabolic therapies easier. There's a huge amount going on. A lot of it we can test and taste outside. The dietitian support is not only critical, but the problem is not all cancer patients, in fact, most of them don't have access to appropriately trained dietitians. The other problem with that is the dietitians that are trained, that are registered dietitians, might not be trained in ketogenic diet, and they might not be trained in ketogenic diet for cancer patients. Because if they don't have some cancer dietitian and ketogenic dietitian, it's not necessarily the same as epilepsy and ketogenic diet. The ketogenic diet part's the same, but some of the side effects and things they have to worry about when you add in chemotherapy or radiation, those are things that could change. This is starting to happen, the recipes, affordable pre-made meals. The pre-made meals are good, but they're expensive. And it's not because these companies want to gouge anybody, it's because they're expensive to make. So if we can get them accepted as a medical food, the insurance companies will pay for it. That will completely change the face of this, I think. The physician buy-in I mentioned before is absolutely critical. We've got to dispel their false concerns, and I think I've heard that already this morning, is first of all, weight loss, fat loss versus cachexic weight loss. Brain tumor patients do not have cachexia or muscle wasting, but in general, but other cancers do. I know there's going to be a talk later in this meeting specifically on that. But the ketogenic diet is probably muscle sparing, so it actually will probably help reduce this. Physicians think it'll exacerbate the side effects. The patient comes in and says, oh, I'm so tired, and the first thing the physician will say is, get off the diet. It's like, no, any patient getting radiation to the head is tired, okay? So don't blame it on the diet. In fact, the diet might help reduce that, and I know of a few patients and a few people that say, yeah, it made a big difference. Again, physicians who remember this diet from way back when, it's disgusting, it's hard to follow, it will reduce quality of life. Wrong. And that the patient can't do it on steroids. Totally wrong. They might not get their glucose to the 70s if they're on steroids, but they'll sure as heck get it lower than it would have been without it. And it is absolutely doable. I think we need biomarkers. If you want to get this funded, get a clinical trial funded through the NIH, they require biomarkers. So blood glucose and ketones is a good marker. In our clinical trial, we said everybody had to get the blood, not urine. But we don't know what numbers are needed for efficacy. It's probably going to be different for different people and different tumors. And by different tumors, I don't mean glioblastoma versus medulloblastoma. I mean glioblastoma in Jane versus glioblastoma in Joe. These tumors are incredibly heterogeneous. Are the blood values representative of the tumor values? We just heard a fantastic talk looking at things that you can possibly start to think about in terms of imaging to help kind of get some ideas on that. Metabolomics, are there profiles, and you can get metabolomics done on blood, are there profiles that correlate to efficacy? MR spectroscopy is a way to look at biochemistry in the tumor, but a lot of places can't do it. And exactly where they do the analysis is important, and exactly how they analyze the analysis is very important. So I think this is possible for the future. Right now, it's a little more problematic. PET scans, many places can't do that. Glucose is the standard. The other metabolites are not, at least not yet. And the possibility of new scanning modalities, that's a big push in the National Cancer Institute and in the NIH about new ways to scan, relatively non-invasive ways to monitor just about anything. And I think we need to really be thinking about it for our kind of work. I think that the ketogenic diet can fit in with the personalized medicine thing. The whole problem with personalized medicine, in my very humble opinion, is when you look at tumors, they're heterogeneous. That's a fancy word for different genetics. You take one tumor from one person, and you look at the cells near my thumb, and you look at the cells near my little finger, and they are not the same. Okay? They are different genetically, they have different capabilities, these tumors constantly change. Personalized medicine takes a sample of one part of the tumor, usually, because that's expensive, analyzes it, finds a marker, and then treats based on that. And yes, that is helping, because it'll get a lot of the cells, but it doesn't get them all. Altered metabolism is kind of present in all these tumor cells, so why the heck don't we capitalize on that? I think if we do start to look at the molecular and biochemical data from tumor tissue from patients that have been on a diet, we can get a huge amount of data, and we're actually working on getting some funding for this. You can use molecular profiling. The same way they do it for any other drug, why don't we do it for metabolic therapies and see maybe if we can come up with patients that do or don't respond. And there have been some work, Ken Schwartz and some others have actually been looking at certain enzymes that might predict this. What about the molecular biology of the tumor? Some tumors will work, some won't. There's a recent paper that tumors with a particular mutation in BRAF are not going to work with a ketogenic diet. It's just one paper, but these are things we have to look at. What about when the tumor comes back? Will this help? We've got evidence from our data in the lab that if we take cells that are resistant to therapies and we add ketones, we can resensitize them. This is really good for tumors that recur because they're usually resistant. Again, adding metabolic therapies to other therapies, based on the molecular biology and the mechanisms that we've done and that others in this room have done, I don't see a reason why this shouldn't be an adjuvant to just about everything, because I think it's going to make just about everything work better. And with that, I'm going to end. Acknowledgements, my past and present lab members. The stem cell work was done by this girl who was a high school student. And actually, some other radiation work was done by Alex when he was a high school student. He's now in college. My grad student, Eric Wolf, has done a great deal of this work. Previous people in my lab. My collaborators, I absolutely couldn't do it without them. And for funding, I'd like to acknowledge Janice Nicholson bequest, and now I'm blocking it because I took that slide out by accident. Max Love Project has started to help us, and this is very embarrassing because I'm blocking our other funding sources. Students supporting brain tumor research. It's a bunch of high school kids that raised money for research, and they actually supported this. That's the one I wanted to make sure I mentioned. So with that, I'm going to close, and I'd be happy to take any questions you have.